going through developmental and growth-related changes. Otherwise, there's trouble. Today we're going to talk about the glucostat, means how you keep glucose static or non-changing. Because too much glucose is bad. Too much glucose, you'll develop high blood pressure, which you can die from. So there's a hormone that regulates glucose. If glucose goes up, the hormone insulin responds and insulin goes up. But too little glucose, going fast already. Y'all gotta stop me today. I'm working on about 72 hours with no sleep, so got no control right now. If glucose goes up, your blood pressure will go up. That would be bad. So if glucose goes up, insulin goes up. And the job of insulin is to bring glucose down. If glucose goes up, if insulin goes up, glucose goes down. But if glucose goes too far down, you can go into hypoglycemic coma and die. The brain has no glucose storage. You always have to have glucose in the blood. So if glucose goes down, insulin goes down. And if insulin goes down, glucose goes up. Woo-wee. The dial goes on forever, doing nothing, and yet everything gets done. Your blood sugar right now isn't going up, down, up, down, up, down. Your insulin levels aren't going up, down, up, down, up, down. It's fine-tuned enough that there's no change. There's homeostasis. The whole job of insulin is to make sure nothing happens. By doing nothing, everything gets done. Today we're going to talk about how exactly insulin does that. I learned something a couple years ago. How many people know about the beta islets? How many people have taken Tom Carlson's course? Okay, so how many people know there really are no beta islets? See, I taught that for years. The books even say that. There are islets of Langerhans in the endocrine pancreas, and the alpha, beta, and gamma cells are all within those islets. There are no separate beta islets. Right? People thought, you can raise your hand again, right? That's what people thought. That's what I was always taught. They're all in the same islet. So you learned from George Bentley that the pancreas is two parts, right? Did he go through that? There's an exocrine pancreas that we'll talk a little bit about. That secretes, it's an exocrine, meaning it secretes to the outer body. It secretes digestive enzymes into the into the digestive tract, which is out of the body. But in these islets, these isolated endocrine cells, I need somebody's belt. If somebody can volunteer their belt, give you an extra point on the midterm. Now, don't everybody rush here. Oh, I will need a belt. They're on the beta islet cells. And I'm just going to write insulin as I for today. There are vesicles that contain insulin. Now here's why I always ask that dumb question. Is glucose a hormone? Because glucose regulates insulin. You can synthesize glucose in the body. It travels. Sort of has a receptor, it has action. Glucose is insulinotropic. There's a glucose permease on the cell surface. Called GLUT2. GLUT stands for glucose transporter. There's 7 of them, but one of them is a pseudogene. One of them doesn't do anything. GLUT2 is not regulated. It's always open. And GLUT2 is how the glucose gets into the beta islet. Because glucose is water soluble. It's not like a steroid. It can't just go across the membrane. It needs a channel. When glucose gets above at 5 millimeters or above, in these beta islets, it's converted to glucose 6-phosphate and ultimately to pyruvate, which has 3 carbons, right? It's been a long time since I had MCP 100. And you'd use that to generate ATP. So if glucose gets above 5 millimeters, the beta islet cells start burning it to produce ATP. The ATP inhibits a potassium channel that's normally letting potassium out of the cell. If you inhibit that potassium channel, what happens to that cell? What? You're saying that because I said the wrong thing last time. It becomes depolarized. Right? If you inhibit the potassium efflux, and that opens a voltage-gated calcium channel, and calcium is required. I've told you this a few times before, and I can't find that wonderful video. Calcium is required for vesicle fusion to the membrane. So once that calcium comes in, I'm just going to call insulin I. So glucose is insulinotropic. If glucose goes up in the blood, insulin goes up. Glucose goes in, it gets metabolized, you generate ATP. The ATP shuts off potassium efflux, which depolarizes the cell. The voltage-gated calcium channels open, and that allows insulin to be released. So glucose regulates the insulin. Insulin, in turn, as I'll show you in the next few minutes, will regulate glucose. That which must be diminished must first be allowed to inflate. If glucose didn't go up, insulin couldn't go up. And if insulin didn't go up, glucose couldn't come down.
Could you explain what you mean by 5 millimeters or give a...
What?
Can you explain what you mean by 5 millimeters or give a...
5 millimolar. So when the concentration of glucose gets to 5 millimolar, yeah, millimolar, not millimeters, 5 millimolar or higher, it starts stimulating the insulin to be released. Yeah?
What's the baseline for glucose?
At or below 5 millimeters. I don't know. I mean, 5 millimolar. Got me saying millimeters now. Give me 5 millimeters of glucose. Millimolar. So not above 5 millimolar. Because 5 millimolar is what stimulates the insulin release. And again, this doesn't really happen. Your glucose and insulin don't go up and down and up and down, but they do. It does everything, nothing, and yet everything gets done. Yeah, what's up, Sharon?
How does that compare to its concentration in milligrams per deciliter?
I have no idea. 5 millimolar, you can calculate it. I mean, I can't. What's the weight of glucose? Anyway, we can do that later. I'll make that a special question under midterm. I don't know how it translates. It's pretty easy to figure out. Insulin has several targets. The big one is the liver. The biggest place that you store glucose as glycogen is in the liver. So I would talk about that first. So in the liver, insulin binds its receptor. And now this is a little bit complicated, only because I've seen several different things. The receptor, you might recall, I'm going to draw a blow up of it over here. It's a heterodimer. It has an alpha subunit that's completely extracellular, and it has a beta subunit that's a transmembrane protein. IGF-1 and IGF-2 receptors are very similar. The alpha subunit is held onto the cell because there's cysteine disulfide bridges that attach it to the beta subunit that's an integral or transmembrane protein. Does that make sense? So the beta subunit has an extracellular portion, a transmembrane portion, and an intracellular portion. The alpha subunit is not in the membrane attached, but it's attached to the beta subunits. We're good? So here's what I don't know the answer to. I've seen it like this, the insulin binds. Insulin definitely binds the alpha subunit. But I've seen pictures of it like that. That's what I usually see. But if you Google around, you'll also see pictures like that where there's two insulins bound to each receptor. I don't know which one's true. I've always seen it with the one. But I know if you Google around, sometimes you'll see it like that. All from fairly reputable sources. Yeah, Matt.
Are the two alpha subunits connected by the disulfide bonds? I can't quite see it from here.
Oh. Here, let me hide it. Let's see if I can draw it bigger. Oh, does that help?
So is that a yes?
What? Yeah, so the alpha subunits are joined by cysteine, a disulfide bridge, and then these subunits are attached to the beta subunit, also by a cysteine-cysteine bond.
Got it.
Okay. The receptor itself is a tyrosine kinase. So when insulin binds, it activates this enzymatic activity, this tyrosine kinase. So some people say the receptor itself is its own second messenger. And if I get a belt, I'm going to show you part of how that works. So the first thing it gets phosphorylated is IRS1. I think I've given you that before. Insulin receptor substrate 1. I guess there's more than one. Actually, sometimes there's not. And then that phosphorylates a whole bunch of other things. But the first substrate is this IRS1.
What phosphorylates it?
The tyrosine kinase. The receptor itself is a tyrosine kinase. When insulin binds, among other things that you'll see in a minute, it activates the tyrosine kinase. So the insulin binds to the alpha subunit, and a signal is transduced through the beta subunit that activates that kinase. IGF-1 and 2 work the same way. In fact, as you might remember, insulin and IGF-1 can bind each other's receptors. So here's what happens. The liver has a GLUT called GLUT7. GLUT6 is the one that's not a real gene. GLUT7 is also not regulated. You'll see what I mean by regulated in a minute. GLUT7 is not regulated. If your blood glucose goes up, it's above 5 millimolar, then the concentration outside the cell will be higher. And glucose will come through GLUT7 on the liver. That's the place where you store most of your glucose. And will come down its gradient. But the cell will very quickly fill up. And the glucose will stop coming in. Right? As the glucose comes in, it will get more concentrated, and eventually the cell will fill up, so to speak. What insulin does is it activates IRS1, and there's probably a few other phosphorylation steps. I don't know them, you don't have to know them, but ultimately, glucokinase is phosphorylated and activated. I think that's the same thing as hexokinase. Yeah or no? Okay, I'm getting some head nods. I learned it was hexokinase. People now call it glucokinase. I'm going to call it hexokinase. And hexokinase converts glucose into glucose 6-phosphate. Then another enzyme is phosphorylated. Dismutase, which converts glucose 6-phosphate into glucose 1-phosphate. Glucose 6-phosphate into glucose 1-phosphate. And then the enzyme glycogen synthetase incorporates the glucose 1-phosphate into glycogen. So it's turning it into this macromolecule, this carbohydrate, thereby maintaining the glucose gradient so glucose keeps coming in. So if glucose goes up through GLUT2 in the beta islet cells, ATP is generated. ATP shuts off potassium efflux, which depolarizes the cell. The depolarization of the cell allows calcium to come in. The voltage-gated calcium channels then let the calcium in. I just said the same thing twice. And then that allows the insulin to be released. The insulin, among other places, binds to the liver. And in the liver, the tyrosine kinase activates the IRS1, which ultimately phosphorylates hexokinase. And dismutase, those are the main targets. And that allows the glucose to be packaged into glycogen. Who's ever been to Japan? I think it's like those guys on the subway, like when you go to Shinjuku or Shibuya, those guys with the white gloves and pack all the people on the train, right? They keep pushing people back so that more people can get in. I think of it kind of like that. So the glucose is coming in this gradient because it's being packed away as glycogen. Questions? Okay, now another thing happens. Upon binding to its receptor, remember the membrane's fluid. So these receptors are moving around the cells. by binding to its receptor, insulin also causes the receptors to start to clump in the cell. And eventually, the receptors, with the insulin bound, get stuck in something called a clathrin-coated pit. And when they get stuck in the clathrin-coated pit, as soon as it's step one, so eventually, these things get endocytosed. So you have a ball of tyrosine phosphorylase in the cytoplasm. So you have a concentrated, so that's also why it says that this is like its own second messenger. And if somebody had given me a belt, I would show you, because I bought some new pants today and I forgot to bring my belt. I don't have a belt. So if pretend that there's another belt attached to this. Who's got a belt? Oh, okay. You're gonna love this demonstration. So this is a big cell, and what happens is, what happens is the green things on my belt, that's the insulin bound to the insulin receptor. You see the orange things, the tape? Those are my cysteine bonds. I make all this stuff myself. You can't buy this in the shop. The orange stuff are the disulfide bridges, and the purple thing is the tyrosine kinase. Can you guys all see that? So this is like on the cell surface. The green there is the insulin. It's bound, and then it starts to clump. And when it starts to clump, you need a bigger waist. See, I can do this better with my waist. I need to fold it in. When it starts to clump, okay, yeah, you gotta put on some weight. So here's what's happening. It's here, you see, and it starts to clump, and it folds up like this. And did you see that? I was kind of standing in front of it. Okay, so can you see it now? I'm putting my hand up like. Can you see it? And so it starts to clump, and then it endocytoses, and it leaves the membrane. Do you guys see that? And so you have, and this is a sphere, not a circle. So all the tyrosine kinase is on the outside, right? So this thing now is in the cell, acting like its own second messenger. It's going around and phosphorylating hexokinase and dismutase, so that more glucose can come in because it's being packed with glycogen. You see? You got it? So it's kind of like its own second messenger. Then, I'm running out of space myself. Then this vesicle, I didn't forget, I just don't have room on myself. Let's do it over here. Then this vesicle, which is going around still phosphorylating things, ultimately forms something called a curl. It fuses, you guys heard of a curl before? It fuses with another vesicle, and it forms a curl. Curl equals the compartment for uncoupling receptor and ligand, or ligand, whichever your preference. And this is not unique to insulin, what I'm going to show you. What's unique to insulin is the idea that the tyrosine kinase is still active once it gets internalized.
Can you read out what you call that pit?
Clathrin-coated pit. Clathrin is a protein that catches these transmembrane proteins. So it's clathrin. So the receptors move around, but once they're bound, they start to clump, and ultimately they're trapped in these clathrin-coated pits, C-L-A-T-H-R-I-N, coated pit. It's a protein. When they fuse with this other vesicle, which I don't know if it has a name. This curl causes a drop in the pH. So they fuse with these vesicles with low pH. That's what the curl is. Yeah.
What does it say, these small circles?
Low pH. LOWPH. Small p, capital H. I'm waiting today. So it fuses with this low pH vesicle. That causes the hormone, the insulin, to come off the receptor. Thus the name compartment for uncoupling receptor ligand. Then these things split. The insulin gets targeted to a lysosome and degraded. But the receptor, once it's uncoupled, the receptor returns to the surface. Kind of opposite of how the insulin got out. And that's not unique to insulin. Remember we talked about the refractory period? That's a part what's going on there. Once oxytocin binds, you internalize, you destroy the oxytocin, but then it takes time for that receptor to come back to the surface. So all the receptors are recycled that way, as far as I know. So should I go through it all again, or we got it? What? One more time. So if your blood glucose goes up above 5 millimolar, glucose through GLUT2 goes into the beta islet cells. It's metabolized, glucose 6-phosphate, the pyruvate, you had that in MCB 100. You generate ATP. There's a potassium efflux channel that's ATP regulated. The ATP shuts off the potassium, the cell depolarizes, the insulin, the calcium then comes in through a voltage-gated channel, and the insulin then is allowed to exocytose that vesicle. In the liver, the main target of the insulin, the receptor's activated, the tyrosine kinase turns on IRS1, and there's a cascade of events that ultimately lead to hexokinase and dismutase, producing the glucose 1-phosphate that the glycogen synthetase incorporates in the glycogen, so the glucose continues to come in through GLUT7. The insulin's further active because the receptors start to clump. They get stuck in the clathrin-coated pits and are internalized, so that now they have a big ball of tyrosine, or many big balls of tyrosine kinase active in the cell. But you don't want that to happen forever, because if glucose goes too low, you can go into a hypoglycemic coma. The brain has to always have glucose. So ultimately, this big ball of tyrosine kinase fuses with the low pH vesicle, insulin comes off the receptor, so now it's no longer active. That's why the little yellow stars are gone. And eventually, the insulin is sent to a lysosome and destroyed, and the receptors return to the surface. We're good? Questions? Comments?
So in step 2 and 3, when you're bringing the tyrosine kinase and the insulin inside of the cell, is it because the clathrin coated pit is already endocytosing other things, or just when the tyrosine kinase is binding to the clathrin, it immediately makes that come in?
Yes and no. So the insulin receptor, for whatever reason, maybe there's some conformational change, the unbound insulin receptor just continues to migrate through the fluid membrane. The insulin receptor only gets stuck in the clathrin-coated pits once it's bound to hormone. So there's some change in the beta subunit, presumably, that allows it then to get trapped. That doesn't happen when it's unligated, unbound. So once it's bound, but yeah, the clathrin-coated pits are always there. And many intramembrane proteins are regulated that way. That's not unique. That's not unique to insulin. But because you wouldn't want to take away your insulin receptors unless it was bound, because then you wouldn't be able to respond when you have a glucose, when you have hyperglycemia. Others? Yeah.
So you mentioned that aldosterone receptor goes through the same exact mechanism starting from number 1.
I think all the receptors do, because eventually they're internalized. Certainly oxytocin does.
And this period here is the refractory period, right? Like this whole thing.
So one of the thoughts is there's a refractory period because you need to wait for these receptors to be returned to the surface. Otherwise, you have a low or no response. In terms of refractory period, the longer time you get. We good? Okay, there's one other thing. Let's see where I want to draw that. Another target of insulin is growing bone and muscle. And bone and muscle have a different GLUT. They have one called GLUT2. And GLUT2 is regulated. Here's what I mean by regulated.
I don't know.
Yeah?
You know, in your beta islet cells, you said GLUT2 is not regulated,
GLUT2 is not regulated.
and now.
Oh, GLUT4. This is GLUT4. Thank you. In muscle and bone, thanks, Mike. In muscle and bone, glucose transporter 4 is regulated. Here's what I mean by regulated. Insulin binds its receptor. It can do some of the same things that it does in liver. But one of the other things it does is there's a storage of GLUT4 in vesicles. And if glucose is really high, one of the signals, there's a phosphorylation cascade that ultimately leads to more GLUT4 is being inserted into the plasma membrane. So that's what I mean by regulated. In growing muscle and bone, you can recruit glucose transporters. The liver, you don't want to regulate it, because the liver you always want glucose to go in, because that's your major storage place for liver. If the liver fills up, so to speak, then you go to muscle and bone, or the same cascade is also regulated by IGF-1. And if your muscle and liver fill up, then it goes to fat. So insulin regulates glucose uptake in fat, And there it's actually converted into fat, which we'll talk more about on Thursday. So muscle and bone have this added feature. In addition to doing all the stuff that liver does, it's got this glucose transporter that's regulated so that you can recruit more and take up more. The one in the beta islets, you don't want to regulate. The beta islet cells always have to take up glucose. Otherwise, you couldn't regulate glucose. The liver always wants to take up glucose. But muscle and bone, you want to regulate. The brain, you don't want to regulate. The brain always has to take in glucose. There's no regulation. The transporters are always there. And I forget which one it is. We're good? Okay, there's one other step. That's not enough.
Is your muscle and bone burning it immediately, or is it storing it?
If you're growing through IGF-1, then it's burning it, and it's actually also using that to convert, using that energy to build protein. But there are other hormones that come involved there, not just insulin, glucose. So it depends on your hormonal state. So as you're going to see, maybe by the end of the day, my phone's dead, so I don't know how much time we have. But maybe by the end of the day, whether or not you grow into protein anabolism and using that glucose that way depends on your cortisol, androgens, growth hormone, IGF-1, and thyroid hormone. So a lot of how you use that is regulated by different. Kind of looks like armadillo. Let me read you that. Oh, that kind of looks like a beaver. Doesn't matter. It's a dog. Whatever. So don't ask me why, but somebody did the following experiment sometime in the early 1900s. They connected a beaver and a flatfish, and they connected two dogs' blood supply. Don't ask me why, I don't know. So they connected these two dogs, and then they did this experiment. They were trying to figure out what insulin did. They injected dog A with insulin, and dog B's blood glucose went way up. And that's how they discovered there's something more going on to the glucostat. And I'll tell you what it is right now. All right, I'm gonna have to erase this. Turns out that insulin is co-packaged with something called GABA. Stands for gamma aminobutyric acid. Gamma aminobutyric acid, which is a neurotransmitter, right? So insulin is co-packaged with GABA. And every time insulin comes out, GABA comes out. And GABA, in this case, is an important neurotransmitter. But in this case, on the alpha islet cells, which contain glucagon, And the GABA receptor is itself a chloride channel. So when the GABA binds, chloride comes in. And what does that do? This time it hyperpolarizes itself. And that means that the glucagon can't get out. So go back to your glucostat. If glucose goes up, insulin goes up. If insulin goes up, glucagon goes down. The alpha islet, glucagon, can't see how much glucose there is. But insulin, through the co-release of GABA, keeps glucagon from going up. If insulin goes up, glucose goes down. I just showed you why. Because in muscle and bone, you're inducing GLUT4. In the liver, you're inducing glycogen storage. So if glucose goes up, insulin goes up. If insulin goes up, glucagon goes down. And if insulin goes up, glucose goes down. But not really. When the glucose goes down, you stop releasing insulin, right? And if you stop releasing insulin, then you stop releasing GABA. If you stop releasing GABA, then the cell depolarizes, voltage-gated calcium channels open, and glucagon goes up. So when insulin goes down, glucagon goes up. If insulin is yin, glucagon is yang. Looks like they're opposing each other, but they're actually working together. Because if insulin goes up and glucose goes down, you need glucagon, to bring the glucose back up. And here's how glucagon goes up. Glucagon in the liver let me show you something else. That works.